Welcome to Blick's Box. If ancient mysteries and cutting-edge science fascinate you, trust me, you're in the right place. Today, we're diving into an incredibly exciting topic: how DNA sequencing is revolutionizing the field of archaeology. From unearthing ancient secrets to rewriting history, this technology is changing the way we understand our past. So, let's start with the basics. What exactly is DNA sequencing, and why is it such a game changer for archaeology? DNA sequencing is a groundbreaking technique that allows scientists to study ancient remains at the most fundamental level genetically. By extracting and analyzing DNA from ancient bones, teeth, hair, or even soil, researchers can uncover a wealth of information about ancient humans, animals, plants, and even microorganisms that once roamed the earth. Imagine it like decoding the hidden messages left behind by time itself. This isn't just about reading genetic codes. It's about piecing together the story of life that existed thousands, even millions, of years ago. Through DNA sequencing, archaeologists now investigate who lived in a certain region, where they came from, and how they interacted with their environment. This technique has the potential to change everything we thought we knew about the past. In essence, we're now able to bring the dead back to life, not literally, of course, but by understanding their genetic makeup. We're unlocking the deepest layers of human history in ways that were never before possible. But why is this important? DNA sequencing gives us a direct window into the past. It helps archaeologists to go beyond what traditional tools like pottery or stone tools can tell us. Through genetics, we can determine things like a person's ancestry, their health, and even how they lived thousands of years ago. Exciting, right? One of the most fascinating applications is studying human migration. Migration, for instance, DNA from ancient skeletons, has revealed how early humans moved out of Africa, spread across continents, and adapted to different climates. These discoveries help us connect the dots of our shared history. And it's not just about humans. This technique has discovered extinctive species like Neanderthals and woolly mammoths back into focus. Their genetic data provides clues about their biology and the environmental challenges they faced. This information is crucial for understanding how species evolve and sometimes disappear. DNA sequencing is rewriting history in many ways we never thought possible. It's helping us solve age-old raising new questions about life on Earth. As we dive deeper into the wonders of DNA sequencing in archaeology, let's explore some groundbreaking discoveries made possible by ancient DNA or DNA. This remarkable field has reshaped how we understand ancient populations, extinct species, and even our own evolutionary journey. One of the most exciting breakthroughs is the ability to reconstruct genetic relationships between ancient populations. For example, now scientists have uncovered genetic ties between Europe's first farmers and populations in the Near East. This discovery reveals us how migration and cultural exchange shape early societies, giving us clues about how civilizations evolved over time. DNA doesn't just stop at humans. It's helping us identify extinct species and decode their traits. For instance, DNA extracted from mammoth tusks has revealed genetic adaptations for surviving the Ice Age. Similarly, studies of saber-toothed tigers have provided insights into their hunting strategies and habitat preferences. And then there's human evolution, and one of the most fascinating puzzles of all. Ancient DNA has provided us with groundbreaking insights into how our ancestors evolved and interacted with other species. One of the most significant discoveries is that early humans didn't evolve in isolation. Instead, they interbred with Neanderthals and other archaic human species, such as the Denisovans. This is where things get really intriguing. Through DNA sequencing, we now know that these interactions weren't just brief encounters. They left lasting genetic legacies. In fact, about 1 to 2% of the DNA in modern non-African populations comes from Neanderthals. This genetic inheritance has had a profound impact on our species, influencing everything from our immune responses to how we adapt to environmental challenges. For example, some people have inherited Neanderthal DNA that boosts their immune system, helping them fight off diseases that were once common in the past. Additionally, ancient DNA has also revealed how Denisovans contribute to human adaptation to high altitudes. Populations living in places like the Himalayas have genes that can be traced back to Denisovans, allowing them to survive in areas with lower oxygen levels. These discoveries are reshaping our understanding of human evolution, showing that we're not just a product of one line of ancestors, but a mix of several different human species. This gives us a more complex and fascinating picture of how we came to be who we are today. Each discovery rewrites what we thought we knew about our past. DNA has become a time machine of sorts, uncovering forgotten connections between species and shining a light on the factors that shaped human history. But there's still more to uncover. DNA sequencing isn't just about understanding the past. It's also opening doors to questions about how ancient findings impact the present and future. Stay tuned as we unravel even more layers of this scientific revolution. So far, we've explored how ancient DNA helps reconstruct the past and unearthable discoveries. But what's making all of this possible? Let's take a closer look at the technical advances that are driving the DNA sequencing revolution in archaeology. First, there's next-generation sequencing, or NGS, a technology that's nothing short of a game changer. Traditional methods could only analyze small amounts of DNA, and the process was actually slow. NES, on the other hand, allows for high-throughput analysis, meaning scientists can now sequence massive amounts of genetic data in a fraction of the time. This breakthrough makes it possible to work with highly degraded DNA samples from ancient remains, something that would have been impossible just a few decades ago. Another major challenge in working with ancient DNA is contamination. Imagine trying to separate a few surviving strands of ancient DNA from modern genetic material that have seeped in over time. Thanks to advancements in lab technique, scientists can now prevent contamination more effectively. Clean room environments, specialized extraction methods, and advanced chemical treatments ensure that the DNA they're analyzing truly belongs to the ancient sample. Accuracy is another critical piece of the puzzle when it comes to DNA sequencing in archaeology. In the past, sequencing ancient DNA came with its own set of challenges, primarily due to the degradation of the DNA over time. Ancient samples, often recovered from bones or teeth that have been buried for thousands of years, are usually fragmented and damaged, making it difficult to extract clean, usable genetic data. Sequencing errors were common, and even the slight to misinterpretations of the genetic C code, distorting our understanding of ancient populations. However, thanks to advancements in technology, this has changed dramatically. Modern algorithms, sophisticated software tools, and high-throughput sequencing methods have drastically improved error correction. These innovations allow scientists to process the most degraded DNA with unprecedented accuracy. What once seemed like an impossible task, studying ancient genomes has now become much more reliable and efficient. These improvements ensure that the genetic data being studied is as accurate as possible, providing clearer and more precise insights into the lives of ancient humans, animals, and even extinct species. With these refined tools, researchers can now reconstruct ancient populations, genetic makeup, determine their relationships with other species, and even gain insights into how they lived, adapted, and interacted with their environments. This level of accuracy is critical in piecing together the complex story of our shared history. These technical innovations aren't just making ancient DNA analysis faster and more reliable, they're expanding the possibilities of what we can discover. From extracting DNA from previously unusable samples to decoding entire genomes, the progress in this field is truly mind-blowing. And it doesn't stop here. With each new development, we're coming closer to solving the mysteries of the ancient world. But there's still more to uncover. Stick with us as we explore how these breakthroughs impact our understanding of human history and the natural world. Now, let's look at the fascinating ways DNA is being used in cultural and environmental contexts to uncover more about ancient life. Ancient artifacts aren't just objects of historical significance. They can hold genetic clues that reveal much more about past societies. Take pottery or tools, for instance. While they may seem like simple objects, they can carry traces of plants or animals that were used in their creation or consumption. DNA analysis of these artifacts helps us understand ancient diets, agricultural practices, and even trade routes. Take food remnants found in ancient pots or on stone tools, for example. Traditionally, archaeologists would analyze the trace through physical traces, but DNA sequencing has taken this a step further, allowing for a deeper understanding of what early humans ate. By extracting and analyzing the genetic material left behind in food residues, researchers can identify the species that were consumed or processed, providing a rare and detailed glimpse into the dietary habits of ancient peoples. This technique is particularly valuable because it allows us to identify foods that would otherwise leave no trace in the archaeological record. From grains to animal proteins and even plant materials, these genetic markers paint a more comprehensive picture of the foods that sustained ancient populations. For instance, DNA from ancient pottery might reveal the presence of domesticated crops like wheat or barley, while traces on stone tools can uncover the processing of animal fats, bones, or even fish species that were central to survival. Moreover, these insights help archaeologists understand how early humans adapted their environments. By analyzing the variety of foods consumed, can uncover patterns of resource utilization, shifts in diet based on climate or geography, and the ways in which ancient societies interacted with their ecosystems. These findings also offer crucial information about trade routes, migration patterns, and the cultural significance of foods, providing a window into how food shaped not just survival, but social structures and cultural practices. In essence, DNA sequencing has opened up a new chapter in the study of human history, allowing us to trace ancient diets with a level of accuracy and depth that was once unimaginable. Beyond the fascinating insights into human history, ancient DNA offers the opportunity to reconstruct past environments and ecosystems, giving us a clearer picture of the world as it once was. By extracting plant and animal DNA from ancient soils, sediments, or even ice cores, scientists can piece together the intricate puzzle of ancient landscapes, offering a deeper understanding of ecological conditions and how these environments shaped the lives of past populations. This process is a crucial step in understanding the broader context in which early humans lived and evolved. For instance, plant DNA recovered from ancient caves or archaeological sites can provide a detailed snapshot of the types of vegetation that existed thousands of years ago. This information is invaluable because it allows researchers to track changes in vegetation over time, offering insights into climate shifts, the rise and fall of certain plant species, and how these environmental changes impacted human life. By analyzing ancient plant DNA, we can learn how agriculture began, which crops early humans domesticated, and how they adapted to shifting climates and landscapes. For example, animal DNA extracted from ancient sites can offer crucial insights into the wildlife that thrived in these environments. By examining the DNA of extinct species or the remnants of animals that were once abundant, scientists can understand. Similarly, animal DNA extracted what species were hunted or domesticated, and how human activity influenced biodiversity. For example, animal DNA can reveal how the hunting patterns of ancient humans affected large herbivores like mammoths or bison, or how early farming practices led to the domestication of animals like dogs, sheep, and cattle. By piecing together all of this information, ancient DNA helps us see the past not just as the collection of human stories, but as a dynamic interaction between humans and their environment. It reveals how ancient civilizations adapted to changing climates, what resources were available to them, and how they might have impacted the ecosystems they depended on. This knowledge provides a window into the delicate balance between human activity and environmental change, helping us better understand the long-term consequences of our actions and how they might shape future ecosystems. These insights are crucial not just for archaeology, but also for understanding environmental changes over millennia. Knowing how ancient ecosystems functioned can guide our efforts to preserve biodiversity today, offering us valuable lessons about climate change, extinction, and ecological balance. As you can see, ancient DNA isn't just about piecing together the lives of people long gone. It's also about piecing together the world they lived in. And this is just the tip of the iceberg. Stay tuned as we explore even more groundbreaking applications of DNA sequencing analogy. In our exploration of how DNA sequencing is revolutionizing archaeology, we've seen some incredible breakthroughs. But as with any cutting-edge science, there are significant challenges that come with working with ancient DNA. Let's dive into the difficulties faced by researchers and how they're overcoming these problems. One of the biggest challenges in ancient DNA is the DNA over time. DNA, as a molecule, is quite fragile. Over the centuries, exposure to environmental factors like heat, moisture, and even microorganisms can cause it to break down. This is especially problematic in warmer climates where the degradation process is accelerated. For example, a bone or artifact found in a tropical environment might is so fragmented or decayed that it's nearly impossible to sequence. But thanks to next-generation sequencing technology, scientists are developing more efficient ways to recover these highly degraded genetic materials, allowing for more accurate and meaningful data than ever before. Beyond the technical challenges, there are also ethical concerns to consider, especially when it comes to human remains. The study of ancient human DNA often requires handling remains that have cultural and spiritual significance to modern-day descendants. Many indigenous groups and communities grew concerns about the excavation and analysis of their ancestors' remains. Response: researchers are adopting more respectful and transparent practices, ensuring that these studies are conducted with consent and cultural sensitivity in collaboration with the relevant communities. And then there's the issue of incomplete data. Not every archaeological site yields well-preserved or abundant DNA. Many ancient remains are fragmented or contaminated, and researchers often work with small, incomplete datasets. This can limit the scope of the conclusions drawn from these samples. However, with advancements in DNA sequencing technology, scientists are increasingly able to pull more genetic information from smaller or more degraded samples. While there are still limitations, these innovations are pushing the boundaries of what we can learn from ancient remains. These challenges may seem daunting, but they're not stopping the progress. With every obstacle, researchers are finding innovative ways to move forward, bringing us closer to understanding our past in ways that were once unimaginable. One of the most astonishing breakthroughs in the world of ancient DNA research came from a discovery that might sound almost too incredible to be true for a single-finger bone. This bone, found in the remote Siberian cave of Denisova, belonged to a previously unknown group of humans called the Denisovans. Despite their mysterious and fleeting existence, this tiny fragment of bone provided researchers with the opportunity to unravel the complete genome of the Denisovan species for the very first time. This discovery marked a significant milestone in the field of archaeology and genetics. The Denisovans, who lived alongside Neanderthals and early humans, have long remained a mystery to scientists. While we had evidence of their existence from fossilized remains, there was very little known about their genetic makeup, culture, or how they interacted with other human species. The discovery of the Denisovan finger bone allowed researchers to fill in many of these gaps, offering a rare glimpse into the ancient people who lived thousands of years ago but are now long extinct. Through DNA sequencing, scientists were able to map the Denisovan genome, providing key insights into their biological features and relationships with other hominids. What made this discovery particularly fascinating was that the Denisovans, much like Neanderthals, contributed their genetic material to present-day humans. In fact, genetic traces of Denisovans can still be found in some modern populations, especially in parts of Asia and Oceania. This finding suggests a history of interbreeding between Denisovans, Neanderthals, and early humans, highlighting the complex web of interactions among different human species long before emergence of Homo sapiens as the dominant species. Moreover, the sequencing of the Denisovan genome revealed intriguing details about their physical traits and adaptations. For instance, it is believed that Denisovans had adaptations to life at high altitudes, potentially explaining why modern populations in regions like Tibet still carry genetic markers that provide resistance to low oxygen levels. These revelations are just the beginning of what ancient DNA can tell us about the genetic legacies of long extinct species and their influence on the genetic makeup of contemporary humans. The Denisovan discovery is a perfect example of how ancient DNA is reshaping our understanding of human history. What once seemed like a puzzle with missing pieces is now slowly being put together, revealing a far more intricate and interconnected story of human evolution than we could have ever imagined. Another incredible case comes from inside the Iceman, the 5,300-year-old mummy found frozen in the Alps. Thanks to DNA sequencing, we've been able to unlock a wealth of information about his life. Beyond his famous cause of death, DNA analysis of Dintsey revealed details about his health, his diet, and even the parasites living inside him. For example, researchers discovered that Dintsey had a type of bacterial infection and was carrying traces of a long, extinct species of plant in his stomach. His genetic data also provided Dintsey's ancestry, revealing that he was genetically closest to present-day populations in Italy and the Mediterranean region. DNA sequencing has also revolutionized our understanding of ancient migration patterns. For decades, archaeologists speculated about how early humans spread across the globe, but DNA has gained the ability to trace these movements with incredible precision. In Europe, genetic data has revealed how ancient populations migrated and mixed with other groups, reshaping the continent's genetic landscape. In the Americas, DNA sequencing of ancient remains has helped clarify how humans crossed the Bering Strait and settled across the continents. These discoveries paint a more detailed picture of human migration and the cultural exchanges that shape the world we know today. These case studies are just a glimpse into the vast potential of ancient DNA research. By unlocking the genetic code of our ancestors, we're uncovering not just their physical traits, but also the stories of their lives, health, and migrations. And this is only the beginning. As technology continues to improve, we can only imagine what new insights will emerge from the past. One of the most exciting prospects is the potential to combine DNA sequencing with artificial intelligence (AI). As DNA sequencing technologies become more advanced, the sheer volume of data generated is growing at an unmatched rate. AI is already starting to play a significant role in analyzing this data, helping researchers uncover patterns that would be nearly impossible for humans to detect on their own. Imagine an AI system that can not only sequence DNA but also cross-reference it with vast genetic databases, providing deeper insights into ancient populations, migration routes, and even extinct species. The union between AI and DNA could unlock a new era of discovery in archaeology, allowing us to reconstruct past human histories with incredible accuracy. Another exciting development is the expansion of DNA research into areas with poorly preserved remains. While we've made great steps with well-preserved fossils, many archaeological sites present challenges, such as degraded or fragmented samples. New recovery techniques are pushing the boundaries of what's possible. With improved methods to extract DNA from the most delicate or deteriorated remains, researchers are able to study populations that were once inaccessible. From bone fragments to ancient teeth, the future of DNA extraction is opening up a vast new frontiers, allowing us to learn more about our ancestors, even when the evidence is limited. Finally, ancient DNA sequencing could provide valuable insights into the relationship between human populations and the environment. By analyzing the genetic data of plants, animals, and humans from ancient sites, researchers can begin to understand how ancient adapted to changing climates. We could learn how early humans responded to climate fluctuations, such as ice ages or warming periods, and how these changes influenced migration, evolution, and the development of agriculture. This research could even help us make connections to modern-day climate challenges, providing lessons from the past that might help guide us in the future. As technology evolves, so does our ability to piece together the complex puzzle of human history. The future of ancient DNA research is not just about uncovering the past, it's about connecting our ancient ancestors to the challenges we face today. With new techniques, advanced technologies, and interdisciplinary collagens, the possibilities are limitless. Thanks for joining us on this incredible journey through the world of ancient DNA and archaeology. Don't forget to like, comment, and subscribe to Blick's Box for more fascinating explorations of science and the environment. Until next time, keep discovering and keep exploring.